Good afternoon, everyone. My name is Pascal Laurent, and I'm a commercial marketing manager at Oxford Nanopore Technologies. It gives me great pleasure to welcome you all to our Core Lab webinar series dedicated to core facilities and service providers. Here, we will be exploring the latest sequencing application developed by Oxford Nanopore and how they can help your sequencing laboratory address the key demand of the research community. With the support of Oxford Nanopore, you will be able to adopt and implement application quickly, streamline your workflow, and accelerate every stage of the sequencing process. Oxford Nanopore is here to help. We want to help Core Lab provide better answers to researchers' scientific questions, offer cost-effective solutions and faster results, be a valued expert, and an essential resource for the research community. Oxford Nanopore is evolving to build effective partnership with our core labs. The Core Lab program supports the growth of your facility and is a partnership to maintain your genomic technology expert status. We want to help you attract new research projects to your core lab, expand your application menu and offering, accelerate your project and time to completion, and maintain your expert status with the sequencing technology. The aim of this webinar series is to help your core business expand its technical knowledge, provide guidance for sequencing projects, and give you a fresh opportunity to engage with the research community. Our first webinar covered the analysis of human structural variants. Today, we will be discussing the benefit of nanopore sequencing for a range of cancer research applications, and the final webinar will be about the accuracy of nanopore sequencing. So let's get started on our second lab webinar in the series, PromethION24, What You're Missing Matters. I'm thrilled to be joined today by Dr. Stephen Jones, co-director and head of bioinformatics, Michael Smith Genome Science Center, BC Cancer in Vancouver, and my colleague, Joe Whittaker, associate director, global product marketing, platform and devices at Oxford Nanopore Technology. After the talk, Dr. Jones and Joe will be taking part in a live Q&A session. So please add your question into the Q&A box, which you will find at the bottom of your screen. You can also upvote the question you would most like answered, and we will try to get through as many as we can. This webinar is being recorded and will be made available on demand shortly after the broadcast. So to start us off today, I would like to introduce Dr. Stephen Jones. Dr. Jones' research focuses on the computational analysis of DNA sequences and analysis of genomic and transcriptomic data. He had applied next-generation DNA sequencing technology to determine the mutation and rearrangements driving many tumor types. A key goal is to develop bioinformatic approaches to predict the most efficacious therapy for the genetic analysis of patient tumor samples to help guide clinical decision-making. His talk today is Nanopore Sequencing, Driving Research Insight from Neonatal Intensive Care to Cancer. Over to you, Dr. Jones.
Thank you very much. Pleasure to be here, and thank you for the invitation to speak to everyone today. I'm going to be talking about some of the uses that we've had for a long sequencing modality in many areas of genetic study, as the title implies, from actually neonatal intensive care to understanding more fully the molecular underpinnings of human cancer. Just for some background, I'm at the Genome Sciences Center, which is part of BC Cancer, and it's a genome center that's been around for now over 20 years. And so we've been involved in a number of different studies, as you might think, coming from a cancer hospital, from British Columbia Cancer Hospital. We've been involved in many cancer studies, including the sequencing of the first breast cancer, sequencing the first cancer genome that was used for clinical inference and therapeutic decision-making, as well as a number of studies, including the first SARS coronavirus sequence, as well as a number of rare genetic diseases. I'll be talking about how we can apply long read to medical genetics, how we can think about it in a neonatal intensive care setting where rapidity in sequencing and understanding the genetic nature of a patient is important. And we'll get into some of the ways in which some of the unique aspects of of long-read sequencing is the ability to get the epigenetic signals as well, particularly methylation. And I'll show how we've been applying that to understand more fully human cancer and also hereditary cancer testing. This is a disclaimer to point out that nothing I'm going to talk about implies that Oxford Nanopore's technology should be used to treat clinically any condition. So I'll kind of go back to kind of one initial example that we had, because we were approached by a clinician at the Children's Hospital who said that we had a patient who had done previous genetic testing and found that there was some form of rearrangement at the Duchenne muscular dystrophy locus. That hadn't been an issue previously. The woman had given birth to a healthy female child, but was now pregnant again, and the understanding was that whatever this rearrangement or change was at the DMD locus seemed to be in the child, and also that it was a male child. And so there were a lot of decisions to be made in terms of whether the baby needed to be carried to full term by the parents. But the clinician said, well, actually, we really don't have enough information to make a proper decision, and came to us and said, could we come up with a way to quickly, and the window of time, as you can appreciate, was very small. Can we understand more about what's happening to that allele? And so what we were able to do is we were able to get some DNA, and we were able to do long read sequencing and characterize more fully and more accurately what was going on at that locus. So up here at the top, this is the reference allele. This is the wild type allele with the DMD gene just here. And what we are able to show down here is this is the mutant or the variant allele. And what we can see is we clearly do see a rearrangement. We actually see a duplication of the DMD gene. But importantly, we still retain a functional, essentially wild-type copy of the DMD gene as well. And so that allowed us to determine that we had no evidence that the functionality of the DMD gene was in any way impaired. But knowing now the breakpoints and knowing now what we could look for, we were able to do a simple PCR test on the uncle to show to find out that the uncle actually carried the allele and was perfectly okay. So that really proved that the allele was certainly not a pathogenic allele. So again, that was a really nice story in terms of the speed and the ability to get an answer back to very, very anxious parents. We've been kind of using this technology as well in our hereditary cancer sequencing. There's a number of genes that we routinely now test for hereditary cancer, and so we can advise the families more appropriately. Obviously, BRCA1, BRCA2 kind of genes, but there's many other genes that we're now testing for, and some of them come up with non-obvious or a little bit confusing changes in genes. And so some of the things we want to do is say, can we actually characterize those alleles more carefully and more accurately? So here's an example here of what we looked at hereditary cancer locus. And this again is the reference is up here. We do see an inversion in this region, but actually at the same time, we have functional copies remaining of all the genes. So again, this isn't necessarily then going to be characterized as a pathogenic allele. This gene here, for instance, we're able to do long read sequencing to resolve this. And in this case, again, the wild type kind of reference is at the top. And in this case, we do have an inversion, and we do have the truncation, fusion, and loss of protein coding regions from at least 2 genes here. So that becomes quite a pathogenic allele. So again, it's about more accurately being able to determine fairly readily with the longer reads what is the structure and composition and breakpoints of these lesions. So one of the things that we want to apply this to, and we do have funding, and we are embarking on this and have done some trials, is, you know, in the neonatal intensive care setting, where obviously it's a very expensive place to have a child. Obviously, these are going to be very sick babies and infants. And can we actually get the sequencing done accurately and rapidly to improve what is now the standard of care? And what is appealing about the long reads in many ways is it does replace a lot of single and kind of tests that might be done in serial or indeed in parallel. And so getting a genome from, say, a long read platform would replace, you know, obviously the single gene test. It should hopefully replace the exome sequencing. It should find any large rearrangements and replace cytogenetic testing, and etc. And so what we hope to get from the long read is, you know, all the sequence variants. We hope to infer any copy number changes, any structural variants, and importantly, going forward, the methylation status of that genome as well. This is kind of some of the tools that we've been using. I really won't go into this. I mean, obviously, to sequence a genome, it's still, you know, it's routine, but it's not trivial to correctly analyze someone's genome, and obviously you want to do it as precisely and as accurately as you can. So I won't talk too much about this. It certainly is a very rapidly changing software stack as a lot of the developers move from thinking about sequences in terms of short reads to the much longer reads. So I'll give you another example. We had another patient come in, and as you can see from the slide, had certain clearly health endangering, life endangering phenotypes. And so what would typically happen, there would be the concept that it could be caused by an SMN1, SMN2 kind of gene change at that particular locus. And so the hospital would typically do a bespoke test for that. But what we were able to show is by doing fairly rapidly the nanopore sequencing, we were able to show that there was a change at actually one of the genes, which would be a pathogenic change. Now, what I'm showing you here is the fact that these 2 genes, the reason why there's a bespoke test and there isn't you know, kind of a standard exome, doesn't show up in a standard exome, is that SMN1 and SMN2 are actually highly similar, almost identical genes, obviously from a fairly recent duplication in the human lineage. But which is why the bespoke test kind of hooks into just 1 or 2 kind of known polymorphisms that discriminate the 2 genes. And what I've shown here is the reads, you know, aligned at fairly low stringency, and so it shows a lot of reads in both these regions. But if you actually align to the kind of stringency that you would normally want to do, you see that there's actually clear loss of one of the genes. And so that would indicate to us that there certainly was an issue at that locus. But the thing is, we can actually determine this just by doing the standard genomic sequencing and not instigating a bespoke test. And then if we do the kind of copy number assay that one does with sequence reads, you can also using in this case the nanopore reads again, you can see that there's a clear predicted region of homozygous loss on chromosome 5. So that again helps us narrow down very quickly what the issue was with that child. Another child that we had, unfortunately, born without any eyes and multiple other issues, but no known genetic cause or understanding of what was going on, and obviously that may not help the child, but it certainly might help other family planning decisions that the family might make. In this case, we were able to show to the family and to ourselves, obviously, that there was a very clear rearrangement going on the the right arm of chromosome 13, with losses and triplications and duplications, which was also a de novo event, which wouldn't or shouldn't affect any subsequent children. But we can see what's going on here. But what was nice about the nanopore is that you're able to define the actual breakpoints. We're not just looking at regions that are being amplified, but we're actually able to go through with reads and actually put together the entire breakpoints and the duplications and the triplications and the losses that are occurring in that region. We did publish this, and it may be a little bit easier to see in this diagram what we're going on. So this is what would be considered the normal chromosome, and then on the other chromosome, we're seeing A, B, C, D, E, and then another D inverted with a C, and then another B, and then a C, and then we don't actually get F. So that's clearly a very kind of rearranged genome in this case, or rearranged arm of a chromosome. So one thing I want to get at is how we can think about using methylation for this. This is something that we haven't had access to in a routine way. And so some of the things that we've been looking at is how can we kind of exploit this. And one thing that's very exciting about having long reads is the concept of phasing. The concept is that the reads are going to be long enough that you're typically going to be able to encompass more than one polymorphism, and that you're able to join variants and polymorphisms and SNPs together to form haplotypes. And one thing that we can do, because we're detecting methylation, is also detect then the methylation and haplotype the methylation. So we can tell if there are chromosomes where one chromosome is methylated at the CPG island and the other chromosome, for instance, is not methylated. And that is information we really only got for the first time, using long reads. Methylation arrays will just show a lower signal, but by phasing the sequence and the variants and the methylation, you can actually see that one chromosome is methylated and one isn't. What we also showed in that particular paper is, if we consider what our understanding of methylation is through bisulfite sequencing, which is often considered a gold standard. I won't go through the slide in detail, but what we were able to show is that we certainly were convinced that the methylation detection through long reads, through Oxford Nanopore technology, was equivalent, if not better, to what the kind of signal we would see in bisulfite sequencing. But really, the beauty of the approach is really when you can haplotype. So we took a couple of cell lines and looked at differentially methylated regions, so the imprinted regions where we know or expect to see a differential methylation between the two haplotypes. And then if you can look on the top, we through the sequence variants, we can map the one haplotype, and we can map another haplotype. And then when we layer on the methylation signal, right? So red is methylated, and blue is unmethylated. The methylation clearly segregates quite nicely with the haplotype. And it's kind of not often you get experiments that look this kind of nice, but what this clearly shows is that you can haplotype both the chromosomes from a DNA perspective, but also haplotype the methylation that's kind of decorating that DNA. What we were able to do then is come up with an improved map of human differential methylation. People had found in printed regions over the years, and we were able to kind of confirm many of them. We were able to identify some new ones, and so now we've got this map of imprinted regions where you get methylation only if the chromosome has come from the mother, and you get some regions where you only get methylation, or you get methylation or unmethylation if it comes from the father. And so I will come back to that in terms of why that might be interesting from the for the aspect of cancer patients. So we published that revised map a year or so ago. But one thing that's really, I think, exciting is the concept of haplotyping and phasing these regions with the methylation. So here we have, for instance, a mutation, and well, we have the Lynch syndrome MLH1 promoter, or the promoter of the Lynch syndrome gene. And so the patient did have Lynch syndrome, but didn't actually have any protein coding mutations that we could detect. But actually, when we looked at the methylation of the promoter, it was clear that one promoter, well, there's differential methylation, and that one promoter was methylated and one was unmethylated, kind of clearly indicating that one of the promoters was off, was broken, essentially. And so we don't necessarily know why, because but the methylation of the CpG island is indicating that you've essentially got a loss of function allele. And this certainly does allow us to kind of dig into the kind of the dark matter, all the regulatory elements and all the sequences upstream of a gene or downstream of the gene that might be regulating it in cis. And so presumably somewhere there is a mutation that is causing this promoter to be off. But we don't need at this point to know what it is. We can certainly now look for it, but this does tell us that we do have a loss of function allele, which is quite exciting. So in terms of can the nanopore sequencing improve our analysis? So one of the issues that clinicians often come to us is, we've sequenced a patient, a child, typically with what's thought to be a genetic disease. We've got a somewhat pathogenic allele in a good candidate gene, but it's considered a variant of unknown significance. And so it's very hard at that point for the clinicians to still give that a defined diagnosis. And so they still struggle with the kind of, this isn't a canonical allele, we're not 100% sure, we really don't know how to advise going forward how we should interpret that. So one thing that we can look at is the methylation signatures that go along with loss of function. So if we look at this example here, for instance, with a patient with Prader-Willi syndrome, we can just look at the blood DNA, actually, in this case, and we can look at regions and we can plot the degree of methylation in this region. So between here and here, and we can see that there's clearly very high methylation frequency at this point. And then if we compare it with a relatively large number of, quote unquote, normal unaffected individuals, we see a very different methylation pattern. And so that to us is a very strong pattern. If you had a variant of unknown significance, but you also saw this methylation pattern, you would probably be much more inclined to agree that the person was affected by Prader-Willi syndrome. And so this is some of the things that we'd like to do as we do the diagnosis, because there are well-known signatures associated with diseases. I think this is, for instance, if you use Illumina arrays, for instance, in this case, you let's take Weaver syndrome patients, and you can plot the methylation patterns, and the Weaver syndrome patients cluster over here, and then the control and affected individuals cluster down here. So there are fairly reproducible and known signatures that we can use to kind of help in the diagnosis. And the idea with the nanopore sequencing is that we actually get, at the same time as the sequence, we get the methylation data, and so can we use that immediately to help in the diagnostic process. So it's kind of early, very early days yet. I mean, we've sequenced a very small number of patients, but and it may be a bit hard to see, but we've sequenced a patient here with Coffin-Siris syndrome, and if you look at the methylation pattern as determined by the signature, the patient clusters over here, the normals are over here. If you look at Sotos syndrome, a patient with the methylation determined through nanopore clusters over here, whereas the unaffecteds are at the top here. So this is our goal here, is to try and expand these data sets so that we can use nanopore as a way of confirming or helping to confirm a diagnosis in rare disease. Here's another example where in ADNP, we had an affected individual and the methylation from a nanopore clustered over here. And we had an individual with a variant of unknown significance, which clustered over here. In this case, the patient had somewhat similar phenotypic behavior as an ADNP related disorder, but the clinician was really not convinced and spent about half a day going through the literature saying, well, could it be that an allele in ADNP could give this specific set of phenotypes? And he said that had he known that the alleles, the methylation of the patient did not correlate with ADNP, that he would have been able to more usefully spend his time following up on other candidates. So one thing that we've been doing as well in terms of helping with understanding hereditary cancer is to see whether we can improve that process using long reads. So one of the challenges is, and we all probably know this, is the protocol for sequencing a genome is to cut up the genome into very small fragments. We can sequence it, and we can infer to a certain extent maybe different haplotype blocks. Haplotype blocks aren't particularly super long, but there's protocols where you can get longer haplotype blocks. But crucially, even if you determine the haplotypes, you don't know which one came from the mother and which one came from the father. So you've either got this scenario or this scenario, and you don't know which. So one of the things we're trying to do is, can we actually work out and determine which alleles you inherited from your mother and which alleles you inherited from your father, importantly, without using any parental material. So why does that matter? Well, if you, for instance, have a mutation in the SDHD gene and it's pathogenic, if you inherited it from your father, you're very high risk of getting a cancer during your lifetime. And so that means that you typically have to go for very routine screening to make sure that you haven't got any cancers forming, so that they can be treated as soon as possible. So that obviously is a lot of cost to the system also, but also a lot of psychological stress to the patient. If you inherit a pathogenic allele from your mother, then you're absolutely at no further risk or heightened risk than the average person. So finding out who you inherited the allele from is actually very important. And as you can understand, there will be some people in the population where parental material is not going to be available if they were adopted, for instance. And so what happens is that they will just have to undergo the testing, even though theoretically there's only a 50% chance that they carry an allele that's going to endanger them. And again, there's about 300 people in Canada who could directly benefit if we could tell who they inherited the pathogenic allele from. Likewise, in BRCA1, BRCA2 testing, if someone does show up with a diagnosis of cancer and are found to have a BRCA1 or BRCA2 or other hereditary cancer gene, which is proven to be pathogenic, there is huge societal impetus, especially in Canada at least, to help identify as soon as possible other family members that may carry the allele. So what happens is that as soon as we find a proband with the BRCA1 allele, for instance, that we start consenting family members and start to look to see who may also carry the gene. It's actually, not many people kind of realize this, but it's the consenting process is actually very expensive. So the consenting actually costs more than the sequencing. It's a lot of work to consent, especially if it's a large family. So if we could just tell from the initial test which arm of the family carries the allele and would be potentially, or could the allele came from and could potentially be carriers, then that halves the number of consents that you may have to do. And importantly too, it actually changes the risk profile of people on this side of the family so that they may immediately qualify for services that they wouldn't have otherwise qualified for. So there are some important reasons why we may want to know where an allele came from. And so if we go back to this map that we showed, we do have a couple of hundred regions where the chromosome is saying, who it was inherited from, either the mother or the father. And so what we can do is, if we refine our test, we can get the haplotypes. In this case, we can sequence and get the methylation from Nanopore. There's another technique we use called StrandSeq, which allows us to get very long haplotypes resolved. And then by mapping the methylation onto those haplotypes, we can actually then map entire chromosomes or entire haplotypes to whether they came from the mother or the father. And so we've kind of tested that. We've shown that actually if we have a known trio where the answer is known, we can correctly predict over 99% of SNVs or SNPs across the entire genome, whether they came from you inherited it from your mother or you inherited it from your father. So it's really interesting in the way that we can use the methylation and the phasing in particular to give us really improved insights into what's happening. So I published this with my colleagues Intan Schrader and Peter Lansdorp. I think it was late last year, if you want to read more about that. I won't go into this in any great way, but there's all kinds of potential now that we have kind of long reads and we have methylation, and if we apply this to transcriptomes, we can actually build up allele-specific expression. We can actually now map more accurately like the methylation across a genome, but we can also map more accurately which chromosome is actually expressing more of one transcript than the other. I mean, typically you would expect them to be expressing at a similar rate, but you are going to get variations in that, and what the role that plays in disease, obviously there is likely to be some role there, but we're just starting to get these concepts of these expression maps mapped to haplotypes. And one of the things that, if we look in human cancer, one of the things that we want to do, again, because we can haplotype and phase, is a very common issue in human cancer is that, the two-hit hypothesis is, you get one hit in the gene, one copy, and then you get subsequently another pathogenic hit in the other. But if they're a few exons apart, like for instance, we have a mutation in one haplotype in the blue here on the NF1 gene, exon 21, and then we have another mutation in the other haplotype, in the red haplotype here, in exon 27. So you wouldn't be able to join those mutations using kind of short reads, but now that you've got the ability to kind of haplotype and phase it over longer distances, we can actually say, yes, actually this gene is knocked out because we can show that there's pathogenic mutations on both copies, on each of the copies. And likewise again, another gene here, PAL-B2, where we can show again, exons some distance apart, that we can show that there's loss of function at this locus because both haplotypes carry a pathogenic allele. And so historically, we would have just guessed, that would be the theory, maybe the expectation. But again, we find cases where, guessing would have got you an incorrect answer, and precision medicine is all about being precise, right? So here we've got P10, where we have a mutation in exon 2 and a mutation in exon 8. We would have presumed they were on different chromosomes, but they're not. They're actually on the same chromosome, right? So we would have been wrong there had we guessed. And you can imagine, that patient might go into a trial expecting P10 loss and actually, even causing the trial to fail because it was wrongly assessed. And then in just, in the last few slides here, again, what we can do, as I showed with the Lynch syndrome gene, is, in cancer, we can find the mutations in the protein coding regions that we think are driving, but we're not really able to find the mutations in the regulatory regions that are driving very easily. And so in cancer, for instance, in some of our patients' samples, we can see here that what we did is we plotted the BRCA1 mutation we found 3 patients that behaved differently. So this is what the methylation looks like in the blood, and it's typically the rate of methylation is quite low. But if you look in the tumor for these genes, for these patients, the methylation is much higher, and it's much closer to kind of 50%, you say. And so when we actually then kind of look at these regions and we phase them, we can actually see that one haplotype is methylated and the other haplotype is unmethylated. And so clearly we haven't got a pathogenic mutation in this gene, but it's off, right? And that would be a pathogenic event. And so we can show, like for all these examples, we show across the CpG site for BRCA1, for instance, that one of the alleles is actually not functioning. And so that's, I think, very important information as we interpret genomes to be able to get. And if we can get it easily by phasing and having the methylation phased, then I think that's a very exciting route forward for us. Okay, I'm just going to finish here. There's a very large number of people at the Genome Sciences Center that worked on this work, particularly the personalized oncogenomics project. So I'm just going to put these momentarily up while I just hand now the hot seat over to Joe Whittaker. Hi, Joe.
Thank you for that great presentation, Dr. Jones. Before I hand over and Joe takes the hot seat, I will just briefly introduce him. Joe Whittaker is an Associate Director, Global Product Marketing at Oxford Nanopore. The title of his talk today is PromethION24, What You're Missing Matters. Over to you, Joe.
Thank you, Pascal and Stephen, for that intro, and fascinating to see how you've been using Nanopore's technology tangibly in the field. As a quick introduction, my name is Joe Whittaker. I'm the Associate Director of Product Marketing here at Nanopore. I've got the pleasure of, over roughly the next 10 minutes, just giving a quick outro and really explaining our platform, our technology, what you've seen over the past 40 minutes. With Nanopore's technology, you are able to reveal more biology with sequences from native DNA and RNA molecules, enabling you to have a more comprehensive view of, in this case, the human genome. So how do we do this? The platform is built around our core chemistry made of the nanopore, a motor enzyme, and the run conditions which enable us to detect the flow of current through the pore. As you can see there on your screen, on the left-hand side, and on the right-hand side, this is run at roughly 400 bases per second. This unique capabilities enable us to sequence the DNA and RNA directly without using PCR, giving us a greater comprehensive view of the biology of your sample. Also, the direct sequencing potentially means that the bias that comes from PCR doesn't impact your project, as well as the additional insights, which I'll go on in a little bit to explain, giving you the ability to look at base modifications, etc., which are maintained because of this library preparation. Other key advantages of this technology is the electrical readouts that you get from this. It means that we're able to detect real-time base calling and analysis, giving you real-time results, rapid results, and the ability to look at other samples such as infectious disease samples, which you need to detect at the point of care. We often talk about long read sequencing as well when it comes to Nanopore, but really the way I like to describe it is short to ultra long, anything from 100 base pairs all the way up to 4 megabase pairs. So really giving you that range of capabilities dependent upon your sample. Now that you've seen exactly how it works, what are the advantages? I've explained a few there on the bottom in the blue boxes there, but because of this format, and I'm going to go on to describe the PromethION product range in particular, it really enables us to have scalable solutions from small to large formats. And over the past 40 minutes from Stephen, you've seen how exactly the PromethION in particular, the P24, I believe, was used in most of the projects, is used. This core platform, we looked at allele-specific methylation, we looked at cancer genomics, looked at a pairing of origin projects there. But really with one platform, the P24, you can see on your screen there, as well as the P2 integrated on the left and the P2 solo on the bottom, you're able to look at the whole omics, everything from single cell, gene expression, fusion transcript, phasing, assembly, etc., as you can see on your screen there. And how can you do this? So the Nanopore multi-omics core platform is the PromethION, and it enables the versatile and scalable multi-omic studies. Direct RNA, and it provides variant detection and best-in-class methylation. You also have a direct RNA, which was fairly recently released about 4 months ago, enable isoform and methylation just in a single library. We will be releasing further updates in this platform as well, which is quite exciting. For example, releasing the M6A base calling analysis later this year. cDNA as well, being able to look at gene and isoform level expression, plus the ability to detect variants, such as gene fusions, and then finally single cell and spatial omics, providing gene and isoform level gene expression, revealing up to 80% of biology that is missed by short read sequencing. And really what this comes down to is using the PromethION, you can see the PromethION flow cell in the center there, you're able to reveal more biology with less. Whereas previously with legacy multiomics, you potentially were doing 3 library preparations across 2 sequencing platforms and using 3 or even more different data analysis and trying to integrate those via spreadsheets or other ways. With the PromethION platform, you're able to look at all of this information just with 1 library preparation, 1 flow cell, and 1 data analysis. And just got one or two more slides. I previously talked about the various different omics you're able to do with the PromethION platform on in the middle there. But regardless of the omics that you're investigating, the projects that you're looking at, just a quick reminder of the accuracy metrics for across our portfolio. So just picking out a few of these in the interest of time, please do visit our website if you want to find out more information, and you've got a QR code there on your screen if you wish to take out your phone and scan it and look in a little bit more detail. But all of the investigations that you perform are with the highest accuracy. For example, if you were to do a human whole genome sequence, you've got a single nucleotide variant F1 score of 99.8%. You can see in the middle of your screen. The human assembly is actually reaching closer to Q50 accuracy, and then the microbial assembly has reached over Q50 accuracy. And I really wanted to show this as well. This slide, it's a really good publication, again, on the bottom left-hand side of your screen. If you wish to scan that QR code, you can go look at it in a bit more detail. But this author was able to show that the true positive variant calling from Oxford Nanopore is comparable to state-of-the-art short-read sequencing-based methods, as you can see there. In fact, if you look into the bars on the left-hand side of your screen, you see they're slightly higher than the equivalent short-read sequencing technology. The IGV in the center of your screen as well helps visualize the mutation G to A in all reads. This mutation in particular was a de novo mutation, and therefore something that other technologies would have missed. And final point around our accuracy. So what you will find is with nanopore technology, because what I previously mentioned around the various different real-time analysis benefits of it, there are various different modes you can use, whether it's called high accuracy calling or superior accuracy base calling there. You can see hack or sup. We do recommend hack due to the speed that you're able to gain with the hack compared to sup. So that was just a really quick outro. If you do have any questions, I'm more than happy to answer them in a moment, but just leave you with this slide around our PromethION device. I talked a lot about the P24 in the last 8 or so minutes, but dependent upon your project, dependent upon your needs, there is a wide range of products utilizing the PromethION platform across everything from, you can see there, the P2, which you can run up to 208 whole human genome samples per year, roughly, using 2 runs per week with 2 independent flow cell lanes, all the way up to the P48, running up to 5,000 human genomes per year. So with that, thanks for your time, and any questions, happy to answer them.
Thank you very much. Well, for the sake of time, that's all the time we have for today. Thank you again, Stephen and Joe, for joining us today. If you want to learn more about today's topic, you can find more information by the following link on the screen or using the QR code. That brings us to the end of today's Core Lab webinar, and I do hope you have found it useful. This webinar, along with our previous in the Core Lab series, is available for you to watch on demand. So please visit the Oxford Nanopore website to check them out. You can also check out all of our other previous broadcasts, as well as information about our other upcoming webinar and knowledge exchange. If you have further questions about your project or getting started with Oxford Nanopore sequencing, please contact support@nanoporetech.com. We would really appreciate your feedback on this webinar. Please head below to the poll tab next to the Q&A tab, where you will find a quick survey on how you found today's broadcast. This will remain open once the webinar ends. Thank you, and enjoy the rest of your day.